All right. Thank you all for joining today's Space Syntax Lab seminar. It is a pleasure to welcome Dr. Tamara Zaninović and Dr. Mirela Sertić Perić, who will be presenting their work on introducing space syntax in urban ecology research. Their talk highlights an interdisciplinary collaboration between the Faculty of Science and the Faculty of Architecture at the University of Zagreb, which explores the relationship between soil biodiversity and urban spatial structure in public parks in Zagreb, using space syntax as a novel tool for assessing anthropogenic influences in ecological research. And Tamara is an assistant professor at the University of Zagreb's Faculty of Architecture, where she teaches urban planning and landscape architecture. She earned her degree in architecture and urban planning from Zagreb and completed her PhD at the TU Vienna, focusing on streets as heritage. Tamara acquired her expertise in space syntax during a research stay here at the Space Syntax Lab in 2016, and her interdisciplinary work spans public spaces, streetscapes, cultural and natural heritage, and she collaborates across faculties on urban ecology and soundscape research. Mirela is an associate professor at the University of Zagreb's Faculty of Science, Department of Biology. She holds an MSc in biology and chemistry and a PhD in ecology, environmental and conservation biology. Her research centers on freshwater ecosystems, which particularly focus on hydromorphological changes, urban freshwater habitats, and karst environments. She's also engaged in the water, energy, food nexus, teaches urban ecology, and plays an active role in biology education and sustainability-focused curriculum development. And before we begin, if you have any questions, please do put them into the chat, and we will address them at the very end of the session. And with this, I'd like to hand over to you, Tamara. The floor is yours.
Thank you. Thank you for the nice introduction. Hello to everyone. I will give you an introduction on our collaboration a little bit at the beginning, and I will then continue to introduce this spatial context of our research on integrating soil biodiversity and space syntax. And Mirela will later explain the biodiversity setup together with the statistical analysis. And with that, let's continue to our aim. So we started as a teaching collaboration, and from multidisciplinary approach, we are now with this research opening up questions on how to do an interdisciplinary research, and we hope in future to maybe integrate this interdisciplinarity into our teaching as well. Aim is, of course, raising awareness of the importance of multi-habitats in our urban ecosystems and our cities. And everything for us started in 2020 with urban ecology course, which Mirela actually thought of and decided to do in collaboration with our faculty. So as you can see here from the keywords, the aim of the course was actually to be multidisciplinary, to understand, so that students can understand the social as well as ecological and urban as well as landscape design questions and issues in general thinking about urban ecology. Here is a preview of how we did our multidisciplinary until now. We have on urban ecology, which is an elective course in at the Faculty of Science, and on our landscape design course at the Faculty of Architecture, we have common guided tours, for example, where multidisciplinary experts visit and are guests at those walks on locations which students can choose for their project proposals. Throughout the semester, we have lectures from social science, from ecology, from landscape and architectural design, all together from multiple faculties. Students listen these seminars and courses. And of course, at the end, exhibitions are multidisciplinary from both faculties, and these are actually platforms for dialogue between local communities, and local communities often repeat those exhibitions on their own events and connect them with other things that are happening in those neighborhoods. That was a short preview of, let's say, 5 years of our teaching collaboration. Here is actually short preview. What are our results? Only 2 works, one on the right from the Faculty of Architecture and one on the left from the Faculty of Science. Both the same park is in question, but both use research methodology, and they are really research-based, but in a different form, let's say, in a different a little bit approach, but both had this multidisciplinary framework for their work. You can see differences and similarities, and I will continue with the left one, where our student Marija Dadić from the Faculty of Science decided to continue her work, which she did on urban ecology course, about biodiversity of soil, and she decided to actually explore it further in her thesis, and later on it became a research on how to integrate soil biodiversity and space syntax measures to evaluate the anthropogenic pressure in urban parks. And what we decided to do is to explore 3 objectives and hypotheses. One was to see the composition of soil in 2 parks very near the pedestrian main corridors. And we decided to test biodiversity on 3 distances from those corridors. The aim later was to compare the biodiversity with the space syntax measures, and to test or to explore if these space syntax measures can be an indicators of urban intensity. Here is the spatial context. So the 2 parks are in different parts of the city, one on the east, one on the west, and you can see here the distance from the historical urban core. Both, let's say, similar to the size, more than 300 hectares, so a large urban parks, but very different in their positioning and in their natural settings, as well as some other historical elements, which I will explain a little bit more. Here you can see that Park Maksimir is more connected to the Medvednica slopes, Medvednica mountain on the north of the city, while Jarun Park is more settled in the Sava River, near the Sava River, and it used to actually be a tributary to Sava. Maksimir is actually our historical urban park, one of the oldest in Europe. You can see here let's say 2 phases of its development, baroque phase and romantic phase. And our setting, we decided to put on its real main corridor, which is actually the only remaining axis from the baroque phase. So it's the main axis and main allée in the entire park, which connects the main entrance and the vista point in the middle of the composition of this park. Jarun Park is actually a sports center, and it has entirely different story. It was connected to Sava River, but after the large flood in Zagreb in 1964, this area becomes a swamp because embankments were built. And from there, the gravel was extracted, and during those extraction, actually some lakes were formed. And then in 1987, for the Universiada in Zagreb, this became a park and sports center with the rowing track. And that is actually the time when it became a urban park. For our spatial analysis and space syntax, we use the initial model which I developed in 2016 at UCL, but since my research was streets as heritage, at that time I was not dealing with urban parks at this scale, so they were not drawn on that map that detailed. But for this research, we needed to upgrade this. So we decided to upgrade it with the pathways, with all the pathways which are formally there. And additionally, we added some axial lines which represented, let's say, the public rooms in those parks, which were more meadows where people would rest and often be there. And this is the resulting map, so upgraded map with all the pathways. We used axial and segment analysis in our research, and to show you a little bit larger, the image here is Maksimir Park and Jarun Park on integration map for the entire city. You can see that this map detects what I have been in short talking about in historical development. The Maksimir Park as a historical urban park is kind of a bit more integrated. Actually, it connects directly to the 10% integrational core of the city, while Jarun Park is not that close to the integrational core. Here is a small zoom for you to see on orthophoto the layout and the difference between these two parks and its natural setting. I marked on this comparison from the space syntax analysis between the city, the Park Maksimir and Park Jarun, a bit higher values in some of the Maksimir areas. They are mostly connected to integration, and while Park Jarun has a bit longer segment and line lengths, as you can see here in numbers. We are coming now to the locations inside those parks. We had 4 locations where the microhabitats were explored, 2 in Maksimir, 2 in Park Jarun, and the positioning of those locations was the fact that we needed 3 different habitats. So that was the kind of aim to find the best suitable locations for them. On each of these locations, there were 3 micro locations, but for the space syntax analysis, we didn't had axial analysis for all these 3 points, because the location for this biodiversity testing were not on the main route, but at the distance from them. Therefore, here are the micro locations which the data which was took in statistical analysis. Those were for these 4 locations. Micro locations or micro habitats, as we call them, were located as you can see on photos. So the first location was very, very near, almost directly adjacent to the pedestrian corridors. Then 10 to 15 meters in, there was a second point in narrow green zone. And then the third microhabitat was at the point where this green zone becomes a forest, just at that important transition. Here is a Maksimir locations and microhabitats. And now I will give a word to Mirela about how the rest of our studies was done.
Thank you. Can you hear me?
Yes.
Yes.
Perfect. So for this study, we used pitfall traps as our primary sampling method. These traps were installed on May the 3rd last year and remained active throughout the entire vegetation season, allowing us to monitor changes in invertebrate activity over time. trap contents were collected and replaced at approximately 2-week intervals, specifically on May the 20th, June the 10th, and June the 20th. We chose spring for our sampling because it's the period of peak soil invertebrate activity, making it ideal for capturing the greatest diversity and abundance. Namely, in summer, invertebrate activity often declines due to heat and drought. And in winter, many species are either in hibernation or adapted to frost conditions, which limits their detectability. By focusing on spring and early summer, we actually aimed to capture a representative snapshot of the active invertebrate communities. Next slide, please. So after collection, all invertebrate samples were removed from traps, rinsed with water to remove soil particles and organic matter, and preserved in alcohol. And for taxonomic identification, this is how we biologists do it. The samples were analyzed in the lab using a stereo microscope, and all individuals were identified to the family level, which allowed us actually to assess patterns in soil invertebrate abundance and diversity. Next slide. So additionally, for this research, of course, we had to obtain all necessary research permits for from the respective park authorities. And we collected a set of key physico-chemical parameters, which are quite small here, but visible, hopefully. To better interpret the biological data. So physico-chemical environmental data are really very important for tracking biodiversity, not only space syntax metrics we used here. Soil temperature was measured at the surface and at the 10 centimeters depth using infrared and probe thermometers. Soil pH value, so acidity was determined in the lab. Assessing active and exchangeable acidity, so to see soil's buffering capacity and its sustainability and stability under changing conditions. And we also analyzed soil texture and assessed organic matter content, primarily humus, which is critical in supporting soil fertility and structure and organisms. And these measurements are actually also crucial for understanding the environmental background of each site, and they showed clear differences across locations. Next slide. This slide just shortly summarizes the statistical methods we use to analyze our data. We mostly use non-parametric statistics due to the non-normal distribution of community data. We also applied principal component analysis to identify the main variables driving differences across sites by combining physico-chemical and spatial data and metrics. And this actually helped us to visualize how sites cluster based on all measured parameters. And these analyses were actually a basis for the key results, which now follow in the following slides. So this slide shows the results of the principal component analysis, which included 13 chosen measured soil physico-chemical parameters, as well as detected space syntax metrics. So the PCA results reveal a clear separation between the sites in Maksimir and Jarun parks. As you can see along the horizontal axis, which is mainly driven by space syntax measures, Jarun sites are characterized as those with lower values of parameters that indicate spatial connectivity and integration, and higher soil pH values compared to Maksimir sites. So all these variables listed along x-axis are actually with the minus sign lowering towards the right of PCA graph. And the vertical axis of this graph separates the M and A sites from the P and V sites. primarily based on soil temperature differences. You can see that temperature is listed along the epsilon axis. So M and A sites have a lower surface and subsurface soil temperature than sites P and V, which is most likely because these two sites, P and V, are more open and exposed, making them more affected by atmospheric conditions such as direct sunlight and maybe wind. And these patterns actually highlight how environmental and spatial factors together influence site differentiation, providing context for interpreting invertebrate distribution across the parks. So usually in biology research, we mostly use environmental data, so physico-chemical parameters. For me, it was really interesting to see this overlap between physico-chemical data and space syntax metrics. So I found them really, really interesting, and this was the whole new world for me, I would say. Next slide. This is biological data, so I will be as short as possible. On this slide, you can see the number of soil invertebrate families and individuals. So A is for individuals and B part is for families, which are collected across the microhabitats in Maksimir and Jarun parks at different distances from the main pedestrian corridors. The highest total abundance of invertebrates was recorded at site M in Park Maksimir, which alone accounted for approximately 45% of all collected individuals. And in contrast, site P, which is also located in Maksimir, had only 16% of the total catch. And this was really also interesting data. However, when we applied the statistical test, the Kruskal-Wallis test, to compare the total number of individuals across different sites, there were no statistically significant results. And also there were no significant differences in the number of invertebrate families between the sites. We also tested whether distance from the main pedestrian corridors influenced invertebrate numbers. For the total number of individuals, there were no significant differences between microhabitats at varying distances from the corridors. But when looking at the number of families, there was a significant difference, and microhabitat B, that one located in a green zone, in a mown green zone, had a significantly lower number of families compared to microhabitat C, which was located in a zone transitioning into wood, which is actually somehow expected, because if the grass is mown and cut, we would expect lower number of invertebrates than in the woody part. Finally, when comparing the number of families and individuals within each park across A, B, and C microhabitats, there were no statistically significant differences. And these results actually suggest that distance from human movement corridors may influence family level soil invertebrate diversity. Not significantly, but the differences are visible. Next slide. So this is a nice colorful slide where you can see that we see a breakdown of the percentage share of different invertebrate families across both parks. The graph clearly shows that both parks have high biodiversity with many different families present in varying proportions. But some families are more prominent in one park compared to the other. For example, in Maksimir, a higher abundance of ground beetles and springtails were recorded compared to Jarun, likely due to the park's moist soil and more nutrient-rich soil and lower pH value, so lower, greater acidity of soil, which favors these species presence. Regarding differences between different microhabitats, within Maksimir, certain invertebrates such as spiders and wood lice were significantly more abundant in microhabitat C, located farthest from the main pedestrian corridors. In addition, in Maksimir, we observed a significantly higher presence of the family Aleioididae. So this is a type of beetle, also in microhabitat C, which was located in the woody part. Similarly, in Jarun Park, the only significant difference was observed for the Staphylinidae family, rove beetles, where also microhabitat C showed a higher abundance of these organisms. So overall, this graph, I would say, it highlights how both parks are rich in invertebrate diversity, with some differences in species composition influenced by proximity to human activity. And it's also likely influenced by habitat type and physico-chemical parameters. We should not exclude these influences. Next slide. In this slide, you can see the correlations between space syntax metrics and physico-chemical parameters and the abundance of various invertebrate families across our sites and parks. So for this correlation analysis, we took into consideration all sites and both parks, just to see how it aligns in total. So here here are extracted only families which showed significant correlations with shown space syntax metrics and physico-chemical parameters. We use some figures to show you what we are talking about, the animals. So starting with Cochleopidae, gastropod family, we observe a positive correlation with axial connectivity and normalized integration, while showing a negative correlation with normalized choice values, indicating that these organisms tend to thrive in areas with more spatial connectivity, but they avoid areas disturbed by pedestrians. This is how I explained to myself these numbers, if we take into consideration what the space syntax metrics means. This family, along with Isopoda, it's these wood lice, also has some correlation with soil physico-chemical factors. Specifically, these gastropods show a negative correlation with soil pH and organic matter content, and it's a negative correlation, as we can see. So the lower pH and lower organic matter content we have in the habitat, more of these organisms will be present. This is what these correlations mean. If the correlation is negative, then it's the opposite trend. And if the correlation is positive, then it's positively correlated. So both values increase. For the Lithobidae, so this is the second figure, there is a positive correlation with connectivity, but a negative correlation with entropy, space syntax metric, and also axial integration. Lithobidae also negatively correlates with soil buffer capacity, which aligns with their preference for slightly more acidic soils. Hieronomida, so non-biting midges, show a positive correlation with entropy, but a negative correlation with axial integration. And they also exhibit a positive correlation with soil temperature, showing their preference for areas with warmer and moist habitats. As for the wood lice, they have a positive correlation with the axial integration and normalized integration, but a negative correlation with entropy, showing, so I explained it, that they tend to thrive in areas with more spatial connectivity and lower environmental complexity. And isopods are also negatively correlated with soil temperature. So if the temperature is higher, we will have less isopods on this habitat. Looking at Staphylinidae, so the rove beetles and Diptera, these are flies. These families show a positive correlation with line length, so they like long corridors, which suggests that they favor longer and linear spaces. Dipteras are flies, so it's somehow, logical. And these rove beetles are really quick runners, so it's also, it makes sense. And additionally, Staphylinida have a positive correlation with normalized choice, while Diptera are negatively correlated with this space syntax metrics. Finally, Lumbricidae, these are earthworms, also show a positive correlation with humus content, which is aligned with their role in soil aeration and organic matter breakdown. So when we analyze all these interesting data, space syntax metrics such as connectivity, integration, and line length interact with physical and chemical properties of the soil. And we can say, I would dare to say that they influence the distribution and abundance of different invertebrate families. And these patterns provide important insights into how invertebrate communities respond to both urban space structure and environmental conditions. And as conclusions, we can conclude that we confirmed the hypothesis 1 mentioned by Tamara at the beginning. So our results showed that soil invertebrate abundance and diversity increase with distance from major human movement corridors. Hypothesis 2 was partially supported. So while we observed associations and connections between invertebrate distribution and space syntax metrics, we did not find consistent evidence that higher integration, which we linked to higher urbanization levels, correspond to reduced family level diversity. And the third hypothesis was also confirmed. I would dare to say that the structure of soil invertebrate communities combined with space syntax metrics proved to be a useful indicator of urbanization intensity and its ecological effects on biodiversity. Although this study is quite robust and still preliminary, we would I think that we can extract some interesting baselines for further research here. And the last slide, Tamara, as a conclusion. So we think that these preliminary results linking space syntax and biology, ecology, can be a direction in better understanding of urban environments. Syntactical model and metrics enable quantitative description and comparison of urban spaces. And in my opinion, they may serve as a proxy of anthropogenic influence on biodiversity patterns in urban parks. As soon as something is quantitative and can be linked to other quantitative measures, it's helpful. And we, Tamara and I, are really enthusiastic and think positively for the future. Because we convinced ourselves that through interdisciplinary collaboration in teaching urban ecology, we can get inspired and motivated for collaborative scientific research practice with our students and between us. And this would be all, actually. Thank you for your attention.